Hi, everyone. My name is Kevin Lance, and I'm the Director of Viral Vector Marketing here at Unchained Labs. I'll be your moderator, so I'll be the first to say thank you for joining us today. We'll have a Q&A session at the end of today's presentation. To ask questions, all you have to do is click on the Q&A button in the Zoom navigation bar at the top or bottom of your screen and type in your question. When submitting a question, please avoid clicking that anonymous button, and that way we can follow up afterwards with you if we don't get to it during today's session. We will get to as many of them as we can. And now I'd like to introduce Nellis Dennis, Product Manager for Lunatic and Stunner at Unchained Labs. Today, Nellis will talk us through how Stunner gives a full readout on adenovirus titer, empty full ratio, and aggregation in about a minute using only 2 microliters of sample. And now I'll hand it over to Nellis. Nellis, how's it going today?
I am good. Excited to tell everyone about adenovirus.
Cool. Happy to hear it. All right, Nellis, take it away.
Okay. Okay, thank you everyone for joining this talk. Snag spot on adenovirus titers and empty full ratios with Stunner. As you might have just heard, I still have issues pronouncing this, so some people might think, adeno, what am I watching? Am I in the right webinar? And that's pretty much what I get sometimes internally as well. People don't know what I'm talking about. I'm like, I don't know what this guy's talking about. But that's overall wrong. It's adeno, potentially. I'm still mispronouncing it, but please bear with me. I'm doing my best here. So let's talk adenovirus. By most people known as a common pathogen that can, for example, cause mild respiratory symptoms or pink eye. But for anyone here joining, probably rather a viral vector that can be a great solution to fight diseases. Adenovirus is about 90 to 110 nanometers in size. It contains a double-stranded DNA genome and tons of different protein that form the viral structure. Now, something as complex as adenovirus comes with a lot of questions. Is my capsid intact? What is my capsid and genome titer? Is it at the right size? And so on. It requires a lot of different characterization techniques to try to answer these. And we all know the feeling. It was a busy day at work and you're hungry. It's 5 p.m. in Europe here, so I'm getting to that. You just want a snack, some tasty food to fill up your belly. Well, getting to that goal is just a multi-step process. You need to prep and chop stuff up and cook it all. And it overall takes too long, leaving me with a hungry girlfriend. And in the end, your kitchen is just a mess. And that is what I see with current quick titration for adenovirus. It is just messy. UV-Vis is a fast technique, but getting to titers for adeno is a multi-step process. It requires prepping, cooking your capsid, and it just takes too long. And on top of that, you actually don't know what you just measured. Was it actually adenovirus, or is it just a bunch of DNA contamination, for example? So that is where Stunner comes in to deliver answers on capsid titer, empty full ratio, and aggregation with only that 2 microliter of sample in less than a minute. And so there's no need to break down your capsid, and there's no labels or reagents or standards that you need. You just use the sample you have. Now, how does Stunner do this? So you simply add in 2 microliter of your adeno sample into the input well, and it's pulled into the microfluidic reservoir of our consumable. And at this step, your sample can sit up for 2 hours inside the input well, and there will be no evaporation. Once you decide to load your plate into the system, the empty cuvettes will be read first, and that will remove any absorbance contribution of the plastic. And then a small vacuum will be applied. Your sample will go into our microcuvettes for the actual reading. Because our microcuvettes have fixed path lengths, it can make very precise and accurate reads. And here we can see exactly what sample loading looks like with the help of some fluorescein and a black light. In 2 wells, there's already 2 microliter of fluorescein preloaded, excuse me. And as you can see, to load another sample, you just simply add in that 2 microliter and it automatically wicks into the plate. So on the left, you can see those loaded samples, and on the right, you can see the samples after they're being measured with Stunner. You can see 2 diamond-shaped cuvettes, and 2 of those are the cuvettes for 1 sample. What this also shows is that after pumping, all the sample remains in our consumable. So there's no contact between the sample and the actual pumping system and between the samples underneath with each other. So no cross-contamination possible. And so with that tiny 2 microliter sample, you'll get tons of information. Stunner contains 2 technologies to read each sample, UV-Vis, and that's going to be used for concentration overall, and DLS for sizing and aggregation. While we're doing DLS, we'll get the total intensity as well, so SLS intensity data is also read. And our samples can be read from 1 to 96 samples on a plate at a time, all in 1 hour. So UV-Vis can give you that 8 to 60 that I discussed earlier, but our UV-Vis can actually tell you how much total protein is in your sample and how much total DNA is in your sample. And on top of that, you'll get DLS and SLS data as well. And this will all be 30 times faster than a classic adeno 8 to 60 titration. Stunner will give you more than 3 answers from those 3 techniques separately. What Stunner is doing is shown step by step here. First, DLS will identify the capsids, the aggregates, maybe even smaller particles in your sample, and then how intense the overall signal is from each of those populations. So in the middle, that info helps you split up your total SLS signal, your total scattering signal, into what's actually just coming from your capsids. And then that will mean that's my empty and full capsids combined at the right size. And then finally, UV-Vis will come in. You have that information on your total amount of protein, your total amount of DNA, and that gives us a % full as well. And that will be applied to that range you get from DLS and SLS to actually know where you are. You have 80% full samples, and so this is your titer. All to make sure you know your adeno. The left side, so the total blue bar here, is all the protein in your sample. UV-Vis got you the total amount of protein, and that's going to be expressed as how many possible capsids all that protein can make. Because the information is there, Stunner knows it needs X amount of molecular weight for 1 adenocapsid, and you have a total absorbance, so it makes those calculations for you. And then you add in DLS and SLS information, and it will split up that total bar in the light blue bar, which is all the aggregation and free floating protein junk. And then you have the dark blue bar, which is your actual total capsid titer, empty and full intact capsids combined. And we'll do the same thing for the DNA. So that's the green bar on the right. The total height of the green bar is the total amount of DNA expressed as the total number of viral genomes that you can make. Light green will be the extra DNA, and dark green is the number of full adenovirus capsids in your sample. If you would divide dark green by the dark blue, you will get a % full number. We'll also convert that into an empty full ratio for you. So here we're looking at replicates of the same lot of adenovirus 5. It's almost impossible to see, but on the top left, those are actually 4 overlaying absorbance spectra. And on the right, the 4 overlaying intensity distributions from DLS are shown. Stunner will take all that information that it just gathered and generate those titer graphs that we just talked about. So that will show you 4 very reproducible results. And the % full is portrayed on the bottom right in the yellow bar chart, while purple is a % empty, which is basically 100 minus % full. Okay, so let's check out some other samples using Stunner. Here we're looking at a certain adenovirus 5 sample from a certain lot. In this case, with DLS alone, you can already see something's up with this sample. There's a lot of aggregation there that you can see with the dark gray peaks. On UV-Vis, with a normal 260, we think this is a normal result. Protein versus DNA looks a bit skewed, but let's look at all the rest that we can get from this sample. So overall, there's not much DNA versus the protein, which you can also see with a high % empty. If that's what you're expected, great. If not, that's an alarm bell, of course. But for final titers, Stunner has also picked up that messy DLS signal and use it to divide up that protein and DNA bars into what's actually coming from capsids at the right side. And so that's here, practically nothing is there because the light green and the light blue are what is free and aggregated. So UV-Vis alone would just tell you your total amount, nothing more. So you also need the DLS information to get the full story and see what is wrong. If we look at another ADV5 sample, it's a different lot this time. And here it looks good at first sight. Your DLS looks perfect. On UV-Vis, you get a nice clean peak. Protein versus DNA looks normal. But again, at first sight, if we look at the extra results from Stunner, it is clear that DLS-wise, this was all nice and clean there. But from UV-Vis, we see with the additional calculation Stunner does that there's way too much DNA in the sample. And so we have high DNA contamination. And that is something that would be completely invisible with the classic 8-60 titration. On the left, you see the results from 8-60, while on the right, it is Stunner's dark green answer that is your final titer. And the rest is all DNA contamination. Okay, now let's take a look at a use case. The following data is in collaboration with the company Sirion Biotech. We're going to be looking at data from 3 different adenovirus 5 lots, where the construct and the capsid, those are all the same, but they contain different DNA inserts. And so all these adenoviruses were made by the exact same stable process that Sirion has optimized. They're overall using a physical titer, which for now comes from ddPCR, one, to get an idea where they are in their process and to get an idea of what their eventual infectious titer would be. So basically to check if they're on track. And here they would like to use Stunner instead for multiple steps in that process, just to do that quick check to know if there's any batch-to-batch difference. And so if we look at that data, the dotted green line is about where the infectious assay titers would be. And as what is expected, physical titers are a lot higher. But the exciting part here is that for these samples, Stunner and ddPCR show very comparable results. So using Stunner as that sneak peek in their process would work nicely. And the big benefit here for the customer, Stunner generated all this data, which are octuplicates on these 3 different lots in just 20 minutes, while with ddPCR, this will take you hours. So all of this with flexible experiment setup on Stunner, where you can start with our default serotype, but it's also fully customizable. And if you would have other serotypes, that is no problem. This can all happen on the same plate in the same measurement. Stunner doesn't need any standards or calibrations, so you can just measure it all at once. You just click and go. Giving you all these answers in great looking graphs and easy to use software. So in the end, you get all of this, the capsid titer, the genome titer, the percent full, size and aggregation information from that 2 microliter sample in less than a minute. And that is how Stunner will deliver label-free, standard-free, and hassle-free adeno titers. So let Stunner do all the work for you, and you'll end with all the results you wanted. So forget about cooking after this very busy day. Just get that some delicious quick delivery, and you'll reach your goal a lot quicker. You'll have a full belly, you'll have a clean kitchen, and a lot of time to spare. And with that, that was my quick introduction on adenovirus. We'll take any questions now, and this is our email address as well.
Okay, so thank you, Nellis, for taking us through a look at how Stunner delivers complete titer information down to 189 pp per mil, how Stunner checks the size and aggregation of up to 96 samples in an hour, and how especially these can be used to look at adenovirus sample QC. So we have some great questions that have already been submitted, but as a reminder, you can still ask a question by entering it into the Q&A section of your Zoom navigation bar. All right, let me just pull up that Q&A box. All right, so first off, do I need standards for this assay? What do I blank with?
Yeah, so that's one of the great parts. You don't need any standards, you don't need any calibrations or anything like that. You just run your samples and you just blank with milky water.
Okay. Can it be used downstream? So can this assay be used downstream?
Yeah, something I didn't mention is we have full 21 CFR Part 11 software add-ons. We offer IQOQs and anything like that to get you everything you need to go downstream.
Okay, this one's a question. I think we probably talk about the samples that we've run on Stunner already. So it's asking, is it common for adenovirus 5 to aggregate? So maybe talk about what we've seen so far in the sample we've run.
I would say that's will depend on your formulation and exactly what you're doing. What we've seen is from lot to lot, we saw differences there, very clean samples, but also aggregated ones. So Stunner is a great tool to just do that quick check. If you're in process doing something different with your sample, maybe now it's aggregated or not, or different formulation. Oh, now I have aggregation versus not. So I can't say if it's common or not, but Stunner would be the best tool to just quickly identify when it does happen.
Yeah, and see across the samples that we've run, wide distribution behaviors. Okay, so this is kind of a two-parter. It's more getting around the idea of what does Stunner need to know to run the Adenopon assay. So what information, for example, genome size or protein sequence, is needed as input for the software?
Oh yeah, that's by default we have a general adenovirus preloaded. What that information contains is all the proteins that form the capsid and their molecular weight, and the total DNA that would be in your sample, so your total DNA size. So if you would have something different to begin with, maybe a different DNA, you just load in the total amount of DNA in basis in the software, and then all the calculations happen accordingly. If you really would have a custom serotype or make something completely different, you would need to adjust the different proteins, upload their amino acid sequence, and then the software will do all the rest. It will calculate how the spectrum looks like, what the total molecular weight is of your capsid, and anything like that. So, but basically, what your capsid is and your DNA size overall.
Yeah, I think important that you can save it too, so you only have to do this once.
Only once, yes.
All right, this is related to that previous question. So if you do have, the custom adenovirus serotype, do you need standards or calibrations in that situation?
No, so that's again the great system. It will, based on the information you put in, if it's custom or anything, you just enter, for example, the different amino acid sequence, and the software will calculate exactly how that looks like. And do all the rest there. So you don't need any standards or calibrations. It just runs as is. You just need to tell if it's something different. But
okay. And what about the lower limit of detection?
So we're using two different technologies here. If you're purely with DLS and SLS, the moment you have something higher than 1E9 capsids per ml, we'll give you a titer range, we'll give you size and aggregation information. The moment your sample reaches 5E10 to the 10th capsids per ml, we'll also pick up that UV-Vis signal. So that will give you a protein titer, a DNA titer, and a % full as well, on top of the sizing, the aggregation, and the DLS information.
So there's one question, just clarification. Was fluorescein used in any of the measurements, or was it just for visibility in that video explaining the chip?
Yeah, so that was purely for visibility to make it nice and clean, because water or a general sample you can't easily see. But in none of the experiments fluorescein was used. We don't need that.
Okay. Here's a question related to purification, I'm guessing probably by ultracentrifugation. So can you test samples in iodixinol? Here specifically, we're talking about 40% iodixinol. Maybe you could talk about that topic.
That's a very high percentage, I would say. For DLS and SLS, iodixinol doesn't matter. So you'll get the titer range, you'll get size and aggregation information from DLS and SLS. For UV-Vis, iodixinol is very high absorbing. So there, with 40%, you wouldn't get a result there. Stunner will tell you how much iodixinol is in your sample, but that would be it. So it would need to get out of there lower than 0.01%, I believe, to actually get a good absorbance measurement. But your titer range, your size and aggregation from DLS would still be there. So yes, you can test that.
Yeah, so I think that's important that you still do get that titer answer and that aggregation answer, which is very helpful. Okay, so this is about the consumable plate. So can you use that plate over and over, or is the well one-time use? Can you just go into detail on that?
Yeah, so the Stunner plate has 96 wells in total. It's SBS plate compatible, so liquid handlers and all can load it. And you don't need to run it completely at once. So you can use 1, 2, 15, whatever amount of samples you want. And then next time you can run the rest or in 3 times, 4 times. So it's as many as you need and then use the other ones. But the sample position can only be used once.
Kind of like every other regular old plate out there.
Yeah.
Okay, what other viral vectors can Stunner analyze?
So we have a very similar application for AAVs that will give you empty full ratio, capsid titers, genome titers, all of that. For other viral vectors, we would still have UVVis and DLS information there, so you can get all of that, but we don't have specific applications that would calculate titer for you. By the way, if you would have lentiviral vectors, we have a great other system called Leprechaun to help you with that. So reach out if you want to know more.
Okay, so I guess this question, it's just, is it serotype independent? You could probably talk about, the different ways the software uses the different serotype information.
Yeah, so it will need that information as which serotype it is. If it's really containing different amount of protein, is larger or smaller, larger molecular weight, anything like that. So to that part, it's not serotype independent. You still need to tell the instrument what you're measuring. So it can apply the correct parameters for the calculations. But compared to other techniques where you maybe have a certain kit for a serotype, here we're completely independent of that. So it can run any serotype. You just would need to tell it if what you're running, basically.
Okay. And there's a few questions kind of around this similar topic, looking for some numbers around accuracy and precision for the readouts that it's going to get. So like what's, for example, a typical percent CV that we would see on Stunner?
Yeah, on highly, it will depend on the sample and the purity, of course, but we have had percent CVs of 1.5% over 96 samples on complicated samples like this. So that shouldn't be an issue at all. Overall, lower than 5%, I would say.
Okay. So actually, this is an interesting one. It's just looking for a little bit more about the consumable and the instrument. So maybe can you just describe overall what a simple experiment would look like? We've talked about the plate, we talked about the instrument, just talking through a very quick workflow.
Okay. So basically, when you have your sample and it's homogenized like you would normally do anything there, you just take out 2 microliter, or I would normally run replicates just for having those bit of statistics. So 2 to 8 microliter, whatever. And you just pick and load, right? So you just do 2 microliter, you load it in the sample in the plate. Once you've done that, you put away your pipette, I would say, and then you basically start up your experiment. Very simple flow through the software. It requests the plate, you put it in, and you're done, right? Depending on the amount of samples you measured, it's going to take X amount of minutes, so about 1 minute per sample. 96 samples in an hour, actually, so it's about 45 seconds per sample, and you'll get your data in our analysis software that did all the calculations for you. It's as simple as that.
So speaking of the software, we have a question from a current Stunner user. Do we need to update our Stunner software for this application? How do they get it?
Yes, so you will need to update to our latest version. You can reach out to me directly if you want, to info@unchainedlabs.com, or on our website, there's a software download function, and we'll make sure you get it and you can update accordingly to use this application.
Okay, there's a few questions just around, I would say, Stunner relative to functional assays like infectious titer, plaque forming assays, things like that. Can you speak to that topic, how they differ and how they might fit next to each other?
Yeah, definitely. So they're like any technique that would measure physical titers versus plaque forming units or cell-based assays. It will really depend on the process and the sample if they would correlate. So I can't address that. It's really person to person or lab to lab dependent, I would say. The moment people see correlation, that's where you could find correlation with Stunner as well. But here we're looking at physical titer, not infectious units. So here, what we were showing was physical titers versus ddPCR where you're practically doing the same thing versus infectious titer, which is a different cell-based assay that takes a couple of weeks. So how you would use that in my head at least is for example, just if you're making always the same batch, you measure on stunner once, and in the end you maybe do your infectious assay. Next time you make the same batch, you measure on stunner in process, and then you know you're on the right track. Or you have some signals like something's completely different with this batch, and you investigate before you do all the rest of your workflow, so you can save time and money finding problems sooner, for example.
Yeah, makes a lot of sense. Okay, so I think a lot of the questions we have remaining are either repeats or something that we can follow up on afterwards. Thank you all for the enormous number of questions. It was really exciting to read through all of those. I think if we'll definitely follow up on those after the webinar if you didn't get your answer here today. But like I said, there's starting to get a lot of repeats, so I don't want to rehash the same information. So thank you all for those great questions, and thanks Nels for a great presentation. And thanks to everyone who joined us live today. If you want to have a deeper conversation with our team, please do get in touch with us at info@unchainedlabs.com, or you can learn more from our website, unchainedlabs.com. We'd love to connect and talk science with you. And thank you again for attending our virtual seminar, and I hope you have a great rest of your day.
Bye.